Sir, you can go ahead, sir.
Okay, sir. Thank you very much. Myself, Naresh Babu. I am working as assistant professor in Molecular Modeling and Drug Discovery Division, Ravindra Institute of Pharmaceutical Education and Research, Autonomous College. So today in this session, in the AICTE sponsored the program that is conducted by the Bapatla College of Pharmacy. Now I am going to take the session related to the applications of molecular descriptors. So, which are mostly using to the evaluation of the physicochemical and toxic and metabolism of the different properties of compounds. So we can see, observe that in-silico methods nowadays are playing a very crucial role in the drug discovery process. So we know that when comparing with the in vitro and in vivo studies, the in-silico methods also completely influencing the same priority in the drug discovery, in the drug development process. So in this presentation, I used different material and image from the web sources simply for teaching purpose only, not for the commercialization marketing purpose. So if you observe that the role of in-silico studies in the drug development, so we know that drug discovery is a process where new drug targets are identified. So new drug targets are we're identifying, and novel drug like small molecules are need to synthesize. So with a good therapeutical activity with the least side effects. So in this modern drug discovery, so several multiple tasks we can observe. So where the applications of various these computational tools are very important. So if you observe the drug development in the broad area of drug discovery, so the discovery and development is mainly involved and in preclinical research and clinical development and FDA review and post-marketing monitoring. So many computational methods are available and are complement one another and may be combined to rationalize the drug discovery process. So in this discovery and development process, most of the computational methods are available and those are complementary to one another to get the complete information about the small molecules data. So the different chemical databases are there to get the most of the physicochemical properties and the ADMET properties, and to get that absorption, distribution, metabolism, excretion, and toxicity related of the compounds. So these studies, by using of computational methods, so it is focusing to find out all these to get the lead compound. So candidate compound we are identifying, and then by identifying this candidate compound, then we are going to making a target fishing. So in this we can observe that here also computational methods are involving for ligand based methods or structure based methods or phenotype based methods are completely influencing to make a potential targets. And to convert the hit molecule into a drug, so we need to find out all these parameters, that is absorption, distribution, excretion, metabolism, and safety, toxicity, and pharmacology are very essential. And most of this nowadays we are predicting by using these computational methods or in silico methods we are using. So here we need to know the difference between this hit and drug. So hit, it is nothing but a compound that shows a desired type of activity in a screening assay. So in a screening assay, the compound should possess a desired activity, and this hit drug should convert into drug by obeying this all the physicochemical properties, and it should be converting into a lead compound and finally converting by the different in vitro in vivo studies, the hit molecule is converting into drug. So we know that the drug is a chemical or a substance which has a physiological effect. So when it is consumed or injected into a body, so that is how we can consider it as a drug. So the drug must possess some parameters like safety, efficacy, stability, and it should have the solubility, synthetic viability, and novel mechanism should be present for the drug. So to know this, all this safety, efficacy, stability, solubility, so we should know the physicochemical properties of the drug molecules. So this image taken from the Swiss Institute of Bioinformatics from Swiss Institute. So this picture is indicating that pharmaceutic steps, that is interrelationship between the pharmacokinetic and pharmacodynamic steps. So as we know that pharmacokinetics, so pharmacokinetics are nothing but what the body does to the drug. So you can see here the clear picture given that yeah, absorption, distribution, metabolism, excretion. So all this coming under the pharmacokinetic steps. So after route of administration, either it may be oral or parenteral. So the active ingredient is entering into the body, and what the body does to this drug, that we are going to study in this pharmacokinetic steps. So that is finding of all these properties by ADME. Different tools we can use, in-silico or computational tools. So these, after entering into the particular interaction with the therapeutic site, so site of action and therapeutic or toxic effects we can observe. So this is completely in the case of pharmacodynamic steps. So we can, I given this another image that is interdependence of pharmacodynamics and pharmacokinetics. The pharmacodynamics, it is nothing but what the drug does to the body. So the activity and toxicity, that is in the clinical development to define dose and therapeutic schemes. So in this pharmacodynamics, we are finding the maximal curative effects and minimal unwanted effects we are finding in the pharmacodynamics. And in the case of pharmacokinetics, so absorption, distribution, metabolism, excretion, we are finding to find these parameters to design the drug candidates with a potent and secure, that is indicating that less toxic and more bioavailable. And coming to this picture, that is hit identification and lead identification and lead optimization. So hit identification, so the compounds with desired activity in a screening assay, the compound can called as hit. So as already told in the previous slide, and lead identification, so the compound with the pharmacological activity against a disease that requires structural modification to serve as a drug. So here we can observe the lead identification, the pharmacological activity, the structural modifications is required. So after modification, the lead optimization, the chemical modification of lead compound. So finally, by finding of these ADMET properties, we can find the ADMET properties, we can see that lead optimizing. So sir, is anyone taking that to admit the candidates?
Yes, sir. We are taking care of it. We are taking care, sir.
Okay, sir. Okay, sir. So what is the reasons in the failure of drug development? And one of the study we see in this picture I taken, that is in the study of 198 drugs. So in that 198 drugs, the failure of drug development is mostly happens with the pharmacokinetic properties, the poor pharmacokinetic properties. The prediction of these pharmacokinetic properties are very important in the drug development, as it is becoming a big in this red colored zone. And remaining factors also influencing the drug development, that is lack of efficiency, that is this part, blue color part, that is 30% it is influencing, animal toxicity and adverse effects in man, and commercial reasons and different miscellaneous categories we can observe in the failure of drug development. So what I am focusing in this session is I am focusing completely the what are the chemical databases or ADMET tools are available to know the different physicochemical properties related to the absorption, distribution, metabolism and excretion, and what are the different ranges of those properties that I'm mostly focusing in this session. So first I am going to giving some chemical databases, in-silico chemical databases. So in that first one, that is the NCI. So it is a public web server of CAD, Group of Chemical Biology Laboratory, that is CBL, and also National Cancer Institute and National Institute of Health, NIH. So the institutes like CBL and NCI and NIH. So these are by combining, they develop a NCI database. So this database completely helps to input the compounds in by giving the different parameters, the query type we can give. So NSC number, that is as I told that National Service Center number, and CAS number, that is Chemical Abstract Service number, or formula we can give, or molecular weight range we can give, and by this we can find the similar structures we can find in this chemical database. So just I will show that in the web page. So you can see here, sir, it is visible, sir?
Yes, sir, it is visible.
Okay, sir. So this website, it is indicating that NCI database browser. So in this browser, we can estimate the similar structures in this. So I am taking the simple example by giving the formula. So I'm taking the formula of isoniazid. And giving the options, formula I am selecting, and I am making that no other elements. And by simply making the start search, so it will give the data related to that. Sorry, one thing I missed. So here I need to keep the similarity only with the similarity query. So what the query I given is formula. So given the isoniazid formula, and with that similar query only, I need to get the information. So I make the start search. One minute. So some error it is coming. So actually the similar formula containing compounds it will come, but it is showing different. Some parameters I am missing here. So if you click that similarity search, what is it is given the structures. So individually you can observe the complete data about that compound. So it is going to give the formula and complexity of the compound and its weight, log p values, a number of hydrogen bond acceptors, donors, and rotational bonds. What is the stereochemistry or potential of the compound, that is R and S and E and Z rotations, and composition, that is CHNO analysis it is giving, and Smythe's notation, and what are the different names of the compound. And we can see here they're given that past predictions also given. So this past prediction is giving the probability to be active and probability to be inactive. In the case of all these activities, it is shown. So here we can observe one. So these are the predicted data it is giving in the past. So separately, we can also in the other slide, I given the complete information about the past prediction. So we can go through that later. And next, coming to the next database, that is PubChem. So this is the well-known web server we can observe, PubChem database. So this PubChem database is a quickly find chemical information. From the authorized sources, we can find the summary of the input and similar structured search we can observe and related records. So the chemical names and synonyms, including the IUPAC names and International Chemical Identification Number, that is INCHI. And we can also see summary of the compound. So I given the his search of the compound, that is ibuprofen. So if you observe what is the information we are getting for this compound. So the structure it is in the 2D and 3D it is giving. That is in the ball and stick model, wireframe and space filling and animate form. The 3D. And the names and different identifications it is giving: IUPAC name and IN International Chemical Identifier Number, CAS number, ICSC. So canonical smiles, molecular formula. So all the related information which next we can observe the different synonyms and chemical and physical parameters, the molecular weight, X log P, hydrogen bond donors, acceptors, rotational bonds, so topological polar surface area and complexity. So all the parameters we can observe for that compound. And experimental data also is available if the drug is already marketed in there. So what is the physical properties also we can observe: color and odor, boiling point of the compound, melting point, solubility. So all the relevant solubility of that compound. So ibuprofen I entered, and vapor pressure, log P values, how they calculated, we can find out solubility, log S value, and shelf life, stability, decomposition, and cacopermeability, that is pKa, and next pKa values and dissociation constant. So different data we can observe. We can get the spectral information also, that is 1D NMR, proton NMR, and C13 NMR, mass spectroscopy. And in the mass spectroscopy, we can observe that the GCMS data and MSMS data and LCMS data, EIMS data, IR spectra of the compound, ATR, Raman spectra. So here related records also it is giving. So this database, that is PubChem database, is very useful to find the complete information about the compound along with this safety hazards, toxicity. If you go down, these are the vendors you can observe, and drug interactions clearly we can observe. Pharmacology and biochemistry, and different uses and manufacturing. So complete relevant data we can observe for that. And another database, that is zinc database. This is also widely using. So that is zinc 15. So it is a free database of commercially available compounds for virtual screening. And this zinc contains nearly over 230 million purchasable compounds are there. So here we can see that information: 230 million purchasable compounds are near ready to dock in 3D formats. And zinc also contains over 750 million purchasable compounds. You can search and analog in under a minute, we can observe within minute. So it gives the information like availability stock in different vendors also. So if you make search in these substances, so by entering the name of the substance or by giving the zinc ID or SMILES notation, that is simplified molecular input line entry systems, or international chemical identity number, INCHI. So we can get the information of the compounds. So as we seen in the PubChem, that much of information we cannot get. But the information related to the compound like molecular weight, log P, molecular formula, number of rings, heavy atoms, and activities related to the compound, and predictions based upon the chembel and scaffold of the compound, and clinical trials, what are happened to that particular drug. So clinical trials related we can search in this zinc database. So here I given the search for the ibuprofen. So it is given the information about the ibuprofen, the log P value, molecular weight, all these things. So if you come down in the website, if you observe that, so these are the clinical trials happens towards to the ibuprofen. The complete description of that we can consider, we can observe here. A next chemical database mostly using, that is drug bank. So the drug bank, So drug banks is a pharmaceutical knowledge base that is enabling major advances across the data-driven medicinal chemistry. So here also I am giving the same drug I am giving for finding the different properties. So it is giving the complete description of the compound and structure, its weight and synonyms, and what is the indication, that is pharmacology indications it is giving, and pharmacodynamics related to the ibuprofen, mechanism of action, in clear we can observe, and absorption, the ADME properties and absorption, volume of distribution, protein binding of the drug, metabolism. So how it is undergoing metabolism, it's in a clear reaction pattern it is giving, and route of elimination, half-life of the drug, T-half, and clearance, different adverse effects, toxicity, drug interactions, and what are the different product ingredients. So different brand names of the compound, generic prescription products, brand names. So all this complete information you can observe in this database also, that is a drug bank. So here also you can observe the chemical identifiers, different references, clinical trials. So these are the clinical trials happens towards to the ibuprofen. Dosage form, patents towards to the ibuprofen. So here we can observe the experimental properties like melting point, boiling point, water solubility, log P, log S, and permeability, and dissociation constant, and also predicted properties also we can observe. So these predicted properties like water solubility, as a experimental we seen the same experimental, the predicted properties also we can observe given in this drug bank related to your sets of compound, and predicted ADMET features, that is a human intestinal absorption. It is good absorption, it is having probability, plus it is indicating blood-brain barrier, Kakatu permeable, and glycoprotein substrate or inhibitor, and different cytochrome P415 induction or inhibition, and aims test to find out the mutagenicity and carcinogenicity, biodegradation, rat acute toxicity, that is LD50 finding, and hERG inhibition. So all these ADMET properties also included in this drug bank relevant to your drug. What is the search you given? So this ADMET is given by taking from the ADMET SCR. So from this database, they included all these ADMET properties. And as like in the PubChem, here also you can observe the spectra of the compound we can observe. And coming to the next database, that is Campbell. So another frequent using of database. So Campbell also, it is a manually curated database of bioactive molecules with drug-like properties. So all the drug-like properties you can observe in this database. And it goes together with the chemical and bioactivity genome. So if you give the search for the compound, so it will give the related compounds also. So I began the ibuprofen, the related search, related structures it is represented, nearly 72 records it is shown. So all are related to the structure of the ibuprofen. So similarity structure. So if you are clicking the individual one, so what we get that 74 in the search. So here we can observe the name and classification of the compound. So what are the sources, alternative forms, and molecule features we can observe here. So there's a drug type, whether it is small molecule or large molecule or synthetic or natural, and rule of 5 it is obeying or not. So it is obeying that rule of 5, that is Lipinski rule of 5. So in the further slides we can observe that also. And first in class, and chirality, and prodrug or not, and orally it is takeable or not, parenterally, and topically we can apply this drug or not. So this is the data it is given, and drug mechanisms it is representing drug interactions and clinical data, similarity compounds related to the structure of ibuprofen and metabolism. So the metabolites also we can observe here by the cytochrome P450 enzymes. So 5-hydroxyibuprofen and 2-hydroxyibuprofen initially by the hydroxylation, and further these are undergoing 3-carboxyibuprofen. So complete metabolic process we can observe for the given compound, and activity charts that is giving bioactivity summary and assay summary and target summary of your drug target, and literature related to your drug, target predictions. So what are the target predictions? So coagulation factor, carbonic anhydrase, angiotensin 2 type, and neuronal acetylcholine receptors, and prolyl endopeptidase. And different calculated properties also given, as like we've seen in the previous databases. So like molecular weight, rotational bonds, polar surface area, hydrogen bond acceptors, donors, rule of 5 is accepting or not. So all these chemical properties. So this is a database that is Chembel, and binding DB. So it is additional another chemical database, binding database. So it is a public and accessible database measured binding affinities. So we can find the binding affinities focusing chiefly to the interactions of the protein, considering with the drug targets. So in this database, we can observe that it contains nearly 41,328 entries we can observe, each with the DOI containing 2,96,653 binding data, and different protein targets are available. And we can see the different protein interactions we can observe in this binding protein data bank. And coming to the next, that is ChemID Plus. Sorry. So it is another tool. So it is another tool you can observe, the ChemID Plus. So in this also we can observe the same physical properties we can observe as like we seen in the previous databases, and there is no specific additional features we cannot observe. And protein data bank, this is a well known to all, that is protein data bank. It is giving the complete information about the proteins. It provides the 3D structure of the data, and for the large molecules, that is proteins, DNA and RNA. So these are the molecules for life found in the organisms in the planet. These 3D structures we can utilize for the different molecular docking studies or molecular dynamic studies we can utilize. And NCBI, that is National Center for Biological Information. So it also provides the complete information about the proteins, not to the drugs. So these are the different chemical databases which were useful to find out the different physicochemical properties of the compound, which are useful in the studies, drug discovery studies. So next, coming to the in-silico ADMET studies, that is ADMET tools. So specifically ADMET tools only we can observe. So what are the ADMET properties, prediction, advantages? So the advantage, if you observe, nearly 40% of drug candidates fail in the clinical trials due to the poor ADME properties. So these late stage failures, so the clinical trials, if the failure is happens due to this ADME, so it is going to make a more effect in the cost of the drug development. So the ability to detect these problematic candidates early can dramatically reduce the amount of wasted time and resources and streamline the overall development process. So this accurate prediction of ADMET properties is prior to expensive experimental procedures. So this is the alternative way before making the experimental procedures, that is expensive experimental procedures. It is need to predict the accurate ADMET properties such as HTS, that is high throughput screening, which will eliminate the unnecessary testing of compounds that will ultimately fail. So if you are knowing that before experimental procedure, if these are not having these properties, there is no need to further going for the experimental studies. So in the prediction they are failure. So the ADME prediction can also use to focus the lead optimization efforts to enhance the desired properties of a given compound. So finally, you can see this incorporation of these ADMET predictions So we will make a more part in the development process and generate a lead compounds that likely to be satisfactory in the clinical trials. So this is the ADME properties. Mostly we are estimating in the different ADME tools, that is basic physicochemical properties, absorption properties, that is Caca2 cell permeability, P glycoprotein inhibitor or substrate, and human intestinal absorption and bioavailability and distribution, volume of distribution, protein binding, and blood-brain barrier permeability, and metabolism related to the cytochrome P450 enzymes, and excretion we can observe the clearance and half-life, and in toxicity we are finding the different toxicities in the different tools, not only mentioned in this slide. So different other toxicity also we can estimate by using the different ADME tools. So to use our drug structure in the different tools, we require the different formats you can observe, that is MYL format, HDF structured data file, or PDB format. So we can convert one file into another file by using this Open Babel tool. So this tool will helps to convert any format to any format. So it is freely available. You can download and we can get the Open Babel to convert our files. So here I given the link also I represented to get the tool. So before entering the ADME tools, different ADME tools, first I need to discuss. So I am making discussing of drug likeness rules. So different drug likeness rules are there. So these rules are several experts criterions. So that are used in drug design for how drug like should be present with the different factors representing in the bioavailability. So we can see the different rules there. So one is a well-known rule we are applying to the drug molecules in the drug discovery, that is Lipinski rule of 5, or rule of 5 we can give. So rule of 5 means it is not having 5 rules, so it is having only 4 rules. So all the multiplication with the 5. So that is the one is the C log P, that is not greater than 5, and hydrogen bond acceptors HBA, that is not more than 10, and molecular weight should be present less than 500 Daltons, and hydrogen bond donor should be present not more than 5. So these 4 rules coming under the Lipinski rule of 5. And if you see the Gauss filter rule, so here we can observe the log P should be present between minus 0.4 to 5.6, and the molecular weight considering in this rule is 160 to 480. And in case of molar refractivity, MR, that is 40 to 130, and number of atoms present in the structure should be 20 to 70, and polar surface area, that is topological polar surface area, that is 140, that is it should be present less than 140. So these 5 rules comes under the Gauss filter rule. So if you see the another rules to the drug-like properties, Weber rule. So in these 2 rules, we can observe that is number of rotatable bonds should be less than or equal to 10, and topological polar surface area less than or equal to 140. And if you come to the rule of 3, so here completely multiple with the 3. The rule of 5, that is multiplying with the 5. So in this rule of 3, so you can observe the log P, that is E log P should be present less than or equal to 3, and hydrogen bond acceptors, that is less than or equal to 3, and molecular weight fragment should be present less than 300, and hydrogen bond donors less than or equal to 3. So if you observe some other rules, that is opera rules or MDDR like rules. So here you can see that rules containing so opera rules, that is the number of rings should be present 3, less than or equal to 3, and ring bonds less than or equal to 18, and number of rotatable bonds, that is the rigid bonds should be present less than or equal to 18, and rotatable bonds should be present less than or equal to 6. And Verma's rule, that is you can see molecular weight less than or equal to 500, and topological polar surface area less than or equal to 125, and log D, that is the distribution, it should be present minus 5 to 2, and hack that is number of hydrogen bond donors and acceptors. Hydrogen bond donors and acceptors less than or equal to 9. Number of rotational bonds should be present less than or equal to 12. And another rule, that is Bayer filter or Muge Bayer filter rule. So here we can observe the 8 rules are there. So one is molecular weight, it should be present between 200 to 600, and X log P, it should be present -2 to 5, and TPSA, that is topological polar surface area, less than or equal to 150, and number of rings should be present less than or equal to 7, and number of carbons, it should be present greater than 4, and number of heteroatoms should be present greater than 1, and number of rotatable bonds less than or equal to 5, and hydrogen bond acceptors less than or equal to 10, and hydrogen bond donors less than or equal to 5. And another last rule, different rules are there. So some rules are represented here, and last one is the CMC 50 like rule. That is log P should be present 1.3 to 4.1, and molar refractivity 70 to 110, molecular weight 230 to 390, and number of atoms in the molecule should be present 30 to 55. So these are the different drug likeness rules you can observe. And coming to the next, ADME tools. So in this link, what I represented here, the ADME tools, most of the ADME tools are available in this link. So just I will show that link. So this is the link what I given in that slide. So that ADME tools, that is a VLSD.com, VLSD3D.com. So it is a directory of tools and databases collected over 20 years. So the ADME and physicochemical predictions and related tools we can observe. So all the different tools, links we can observe here. So all these tools we can utilize by clicking the each tool. So different number of tools are there, nearly more than 200 above are there. So if you click any one, it will going to give the particular ADME tool. Here I am representing some, I am giving some introduction to that some ADME tools. So first I am discussing about the tool, that is ADME tool, that is we are using to predict the physicochemical properties, that is Schrodinger OikProp. So in the Schrodinger OikProp, so I clearly explained about these properties in this YouTube link I given here. So you can go through that link to complete information. So here I given the descriptors what we are going to estimate in the OikProp in the Schrodinger. So the stars it is representing. So the stars is indicating that so different parameters are taking and how much of these parameters are obeying. So by basing upon that, they are giving the range. It is they are giving from 0 to 5. So more stars, it is more. It is not obeying. That means it is not having the within the range of these properties. So the star should be present 0. So if the 0 is present means the compound is not violating all these properties. So if it is coming 1, 2, 3, 4, 5 like that, it is given the number. So while predicting in the OikProp, so it is indicating that some of the properties are violating. So these are the properties you can observe in the OikProp. That is amines, number of amines are present, amidine ring present, acids, amide, number of rotational bonds, and CNS permeable, that is activity, CNS activity, molecular weight, dipole moment, and SASA, that is total solvent accessible surface area, and FOSA, that is hydrophobic component of the SASA, and FISA, that is hydrophilic component of the SASA, and PISA, that is indicating that carbon attached to the hydrogen, that is pi component. The sigma pi, that is pi component, that is double bond related. So pi component of the SASA, that is accessible surface area, solvent accessible surface area. So all related to the surface area, that is solvent accessible surface areas. And WPSA, that is weakly polar component of the SASA, that is accessible surface area. And volume of the compound, a number of hydrogen bond donors, so number of hydrogen bond acceptors, and octanol, what is the solubility in octanol, and what is the solubility in water, and what is the solubility in octanol and water, and log S indicating the solubility of the compound, water solubility, And herbicidal inhibition, always it should be present below 5, -5. And caco permeability, caco 2 cells permeability. So caco cells is nothing but immaterial cell line of human colorectal adenocarcinoma cells. And log BB, that is blood-brain barrier permeability, the compound should be present from -3 to 1.2, the range should be present. And QPP MDCK, so this is indicating that predicted apparent MDCK cell permeability. So MDCK is nothing but Madin Darby Cannon kidney cells. So these are taken from the model mammalian cells, and these cells are used to variety of cell biology studies, including cell polarity, cell-cell adhesions, cell-cell motility, and different growth factors also considering in this MDCK cells. And QP, that is skin permeability, log KP. So it should be present -8.8 to -1. And QP log KHSA, that is a prediction of binding to human serum albumin. Human serum albumin. So it should be present -1.5 to 1.5. That is a range should be present to our compound. And percentage human oral absorption. And if it is present greater than 80%, it is high oral absorption. And if it is less than 25, so the compound is having the poor oral absorption. And polar surface area, that is 7 to 200 should be present. And the number of nitrogens and oxygens present in the compound, it should be present 2 to 15, and rule of 5 and rule of 3. So these are the different properties we can estimate in the Schrodinger OikProp. And next tool we can see, that is a Swiss ADME. So here this picture is indicating that Swiss Institute of Bioinformatics having the different tools, that is computer aided drug design tools, that is Swiss parametric and Swiss doc, Swiss sidechain, Swiss growing, and Swiss similarities, Swiss target prediction, Swiss SAR, Swiss bioISTE, and Swiss ADME. So where we are using these completely, they are represented in this figure. So I am going to represent the importance of this Swiss ADME here. So it is useful in the all these three stages: hit to lead identification, hit to lead, and lead optimization, and also in the preclinical development. So this Swiss ADME is going to useful. Not only Swiss ADME, all the ADME tools whatever we are seen. So those are useful in these stages. So you can see Swiss ADME. So it is a web tool that gives a free access to a pool of fast yet robust predictive models for physicochemical properties. Not only physicochemical properties, and also giving the pharmacokinetic, drug likeness, and medicinal chemistry friendliness among with the in-house proficient methods are using in this model, in this tool. So it is a free online tool. Without login, we can directly access this one. So here they are using that boiled egg and ILogP and bioavailability radar. They are using to explain this all the properties. So it is very easy and efficient and user-friendly interface through the login-free website. That is, we can access this by using this link. So the compound what starting from I am seeing that ibuprofen I predicted and I represented here. The properties what we observed in this ADME. So this is the physicochemical properties indicating for the compound. What is the query we given for that particular compound? It is giving the structure, and then it is giving the physicochemical properties, and giving the lipophilicity, that is log P values, and giving the water solubility. These are the water solubility, and pharmacokinetics, that is absorption, blood-brain permeability, and P glycoprotein substrate, and different cytochrome P450 inhibitor or not, and skin permeability, and drug-like properties like Lipinski, Goose, Weber, Egan, Muge, bioavailability score it is giving to the compound. So all these what we discussed in the previous slides. So according to this, it is obeying or not. So in the all these drug likeness, this compound is obeying. That's why it is giving yes, 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 like it is. So if it is not obeying, it is going to represent this no. And pains, that is no, the medicinal chemistry pains it is not there, and bring and lead likeness, and number of violations it is having only, that is molecular weight less than 250. and synthetic accessibility score, it is given 1.92. So that is boiled egg to predict the gastrointestinal absorption and brain penetration of the small molecules. So boiled egg method they included in this ADMED tool. So it is used to know the either it is crossing the blood-brain barrier and gastrointestinal absorption is there or not, that we can find out. So here some drugs I represented: ibuprofen, omeprazole, and some other two drugs which are not marketed, which are simply structures I given. So if you observe that ibuprofen, so the permeability is there. So this yellow color, as like in the egg, yellow color, it is indicating the blood-brain barrier, and HIA, that is the human intestinal absorption, this white region, and this blue color dots indicating that PGP plus and PGP minus penetrating or not, it is indicating. And another, it is indicating the ADMED tool, that is a bioavailability radar. So this bioavailability radar for that ibuprofen I represented here. So this radar is indicating this colored zone, indicating the suitable physicochemical space for the oral bioavailability. So if this radar present inside this red color, so then the drug is having the good oral bioavailability. So if it is coming out from this red color, it is indicating that so it is not having the complete oral bioavailability. So what are the parameters they are considering in this bioavailability radar? Means that is the lipophilicity. It should be present, the compound should have minus 0.72 plus 5, and size of the molecule, that is molecular volume, 150 gram per mole to 500 gram per mole, and polarity, that is topological polar surface area, it should be present between 20 to 130 angstrom square root of angstroms, and insolubility, log S, that is E soluble, it should be present 0 to 6, and in saturation, that is fraction of sp3, it should be present 0.25 to 1, and flexibility of the compound by basing upon the number of rotational bonds should be present between 0 to 9. So if the compound is obeying these parameters, then the drug is having the good oral bioavailability. And Swiss lipophilicity, we know this parameter. So lipophilic molecules and hydrophilic molecules. So difference between the lipophilic and hydrophilic molecules. So lipophilic molecules are well absorbed and access the brain, that means easily crossing the blood-brain barrier, and not released from the albumin, that means it is binding with the proteins, and rapidly metabolized and less excreted by the kidney. So this is the properties you can observe if the drug molecule having the more lipophilicity. If the drug molecule having the more hydrophilic in nature, means it is not well absorbed but suitable for injection and well distributed, so except in the brain, that is it is not going to cross the blood-brain barrier, and metabolically stable but rapidly excreted. So metabolically it is not metabolizing easily, but it is rapidly excreted from the body. So we can observe the optimum, we can observe in this region of the oral drug or drug length should be present from -1 to 5. So it is a region you can observe for the lipophilicity. So this is the prediction of lipophilicity, what parameters they are using in the SwissADME. So completely this blue color in the molecule is indicating that lipophilic, and red colored zone it is indicating the hydrophilic in the 3D structure of the compound. So in the water solubility, what is the formulas they are using to find out that log S value? So historical model they are using, that is log S 0.5 minus 0.01 into melting point minus 25 minus log P. This is the formula they are using. And this solubility in water, it is representing this one, E solubility, moderately soluble or freely soluble or poorly soluble. So this is the quantitative class we can represent in the log S scale. So if it is having -10, that is less than or equal to -10, if it is came, the value. So if you observe here -5.73, so that is indicating that moderately soluble, this region. So 10 to 6, it is poorly soluble. 6 to 4, it is moderately soluble. -4 to -2, it is soluble. -2 to 0, very soluble. And above 0, it is K, means it is highly soluble. So basing upon this, they given the solubility parameters. And next software, that is we can see for ADMET prediction, that is a Stardrop Direct Nexus. So it is Stardrop software. It is a licensed software, so it is not freely available. So in this also we can find out the different physicochemical and different toxicity studies we can find out. So physicochemical descriptors like log p value, molecular weight, hydrogen bond donors, hydrogen bond acceptors. So all these parameters what I represent in this table, so we can estimate. And majorly it is used to find out the toxicological profile of the compound, that is toxicity. TP form is the complete basing upon the whether the compound is toxic or not, all these toxicities. So finally giving a one score to the drug molecule, that is nothing but toxicity. TP form is we are calling it. So what are the different toxicological profile we can observe in this direct nexus software, that is from the Jastra Innovations. So already morning, I think the Girnar Pillais are mostly maybe explain this one. So we can see the different toxicities like occupational asthma, teratogenicity, hepatotoxicity, different toxicities, nearly 70 something the toxicity studies are giving, not only related to the major toxicity, so it is also giving phototoxicity, and it is also giving the Ames mutagenicity, nephropathy, and kidney disorders. So here one good thing is we can observe in this Stardrop Direct Nexus, it is so we can find out the which functional group it is responsible for the which functional group is responsible for that particular toxicity. So for example, I taken a drug that is pazopanib. So it is an anti-cancer drug. So it is a potent and selective multi-targeted receptor tyrosine kinase inhibitor, and it is going to block the tumor growth and also inhibit the angiogenesis. So the structure, if you are observing the pazopanib, so in the structure of this part, this red color indicated part completely producing the hepatotoxicity. So this part is responsible for the hepatotoxicity of the compound. And if you observe this part, so this complete aromatic is not responsible. Some part of the aromatic ring and this sulfonamide group, SO2 NH2 group. So this group is responsible for the ocular toxicity and urolithiasis and bladder urothelial hyperplasia, nephrotoxicity, carcinogenicity, and also phototoxicity. So all these toxicities are related only by this functional group due to the presence of this functional group. So what is the group or scaffold which is responsible for the toxicity we can identify in this tool. So the red color is indicating the responsibility of the functional group or the scaffold or moiety, it is responsible for the toxicity. So one option we can make in this software, we can apply the bioisosterism. So modification in your structure, and we can again predict the either this toxicity is increasing or decreasing. So by the bioisosterism, so by applying of bioisosteric studies, so we can modify the structure and we can find out. So by modification, I make a simple modification by methylation of this sulfonamide group. So I made attachment of methyls here. So and then I observe the toxicity in that same software. So but this complete molecule is not having any toxicity. So by making a simple modification structures, we can reduce the toxicity, and it is helpful to find the structure without any toxic effects. And next tool, that is ADMET lab. So this website link is going to open that software. So these are the different parameters we can estimate by using this ADMET lab. So we can see that in the next slides. So the different rules we can observe, drug likeness rules, what we seen in the previous slides. So all those rules, the obeying or not, we can estimate for our compound. So by giving their SMICE notation, that is simplified molecular input line entry system, or by giving in a SDF format, structured data format. So by giving the file or in the smiles format, we can predict the all the rules we can find out. So the drug likeness evaluation we can do by giving this search. So one drug I given here, that is omeprazole I given. So the molecule omeprazole, so it is obeying 100% matches we can observe to the Lipinski rule of 5. So it is within the range of molecular weight, that is less than or equal to 500. Log P is less than or equal to 5, log P value. And hydrogen bond acceptors less than or equal to 10. So it is 5 only having, and hydrogen bond donors less than or equal to 5. So likewise, we can predict our compound drug likeness properties we can find out. And so along with this drug likeness properties, we can also find out the different physicochemical properties and ADMET properties. So what we've seen in the previous SwissADME and also in the Schrodinger OikProp. So similarly, we can observe. So here the complete given the information about these properties, what is the ranges they are given in the ADMET lab. So log is the solubility, their units you can observe mole per liter. So the suggestion is the molecule should be present greater than 10 microgram per ml, and optimal is higher than -4. So if it is having less than 10 microgram per ml, low solubility, and 10 to 60 moderate solubility, and greater than 60 microgram per ml, it is high soluble. And log D, distribution of coefficient, it is calculated at the pH 7.4, that is at the blood pH, and it is having the suggestions, it is 1 to 5 it should be present. So less than 1 means soluble high, and permeable low by passive transcellular diffusion, and permeability possible via paracellular if molecular weight is less than 200 and the metabolism is low. And if the drug molecule having 1 to 3 and solubility is moderate, and if it is having 3 to 5, solubility is low, and greater than 5, solubility is completely low, we can observe, and the permeability is high and metabolism also high. So basing upon this distribution, what is the effect in the metabolism and also in the permeability we can observe. And log P, this is distribution coefficient, that is should be present 0 to 3. And if it is less than 3, poor lipid bilayer permeability, and greater than 3, there is poor aqueous solubility. And Caga2 permeability, cells permeability, that is should be have greater than -5.5. It is optimal. And P glycoprotein inhibitor, it is categorical, you can observe. So if it is for our compound came 0, that means non-inhibitor. So if it is came 1 means it is inhibitor. And PCB substrate, so same categorize, 0 means it is a non-substrate. If it is 1K means it is a substrate. And human intestinal absorption, so it is also categorized HIA minus and HIA plus. So 0 means minus and 1 means plus. So it is basing upon the greater than 30% human intestinal absorption, less than or equal to, greater than or equal to 30% and less than or equal to 30. So if it is greater, plus it is indicating, that is it is more crossing, more absorbing, less means minus. And bioavailability, that is 20% and 30% is similarly categorized. And plasma protein binding, 90% is significant. And volume distribution, 0.04 to 20, optimal is 0.04 to 20 liter per kg. And blood-brain barrier, so 0 is indicating the blood-brain, it is not crossing the blood-brain barrier, and 1 is indicating the crossing the blood-brain barrier. And here I represented the different cytochrome P450 inhibitors and substrates, and 0 is indicating the non-inhibitor, and 1 is indicating the inhibitor in the result. And next in the half-life, it should be present greater than 0.5. So if it is having greater than 8 hours, it is high half-life. 3 to 8 moderate, and less than 3 hours low half-life. And clearance, it is given in the values ml per minute per kg, and it should be range greater than 50 ml per minute per kg high, and 5 to 15 means moderate, and less than 5 means low. So it is a clearance in the renal. And Herd channel inhibitors, so 0 is indicating it is a non-blocker, and 1 is indicating the blocker. And human hepatotoxicity, so 0 is indicating the negative and 1 is indicating the positive. And Ames mutagenicity, similarly 0 is negative and 1 is positive. Skin sensitization and acute toxicity, that is LD50, so it should be greater than 500 mg per kg. So it is a dose amount of tested molecule to kill the 50% of the treated animals within a given period. So high toxicity if it is 1 to 50 mg per kg, and medium toxicity that drug is having between 51 to 500. So 500 to 501 to 5000, it is indicating the low toxicity. And drug induced liver injury, so it is also categorized negative indicating the 0 is indicating the negative, 1 is indicating the positive. And maximum recommended daily dose, that is categorized 0 is indicating negative and 1 is indicating positive. So by taking one example, I given the values for the what we see in the different parameters in the ADMET lab. So omeprazole I taken, so the log p value I find out that 2.9, and here blood brain permeability BBB, so it is having 0.043. So category it is 0, it is not going to cross the blood brain barrier. And complete data what we seen in the previous slides. So for omeprazole, I predicted and given the data here, and according to the suggestions, it is obeying or not, we can find out here. So log P it is obeying 0 to 3 between it is present, and log D also 1 to 5, 1.0 to 74. And PGP inhibitor, yes, it is PGP inhibitor plus it is came, and PGP substrate it is not having PGP substrate, so negative value. And human industrial absorption it is having, that's a plus, and bioavailability is there. And plasma protein binding it is nearly having 87.19%, and good is 90%. And volume of distribution and blood-brain barrier and different metabolism by cytochrome P450. So it is having the inhibitor and substrate activities. So all are having plus. And except this P450 cytochrome 2C9 inhibitor, it is not having. And also this one also, cytochrome P2D6. And half-life, it is having 1.8 hours half-life. And clearance rate, it is 1.577. So it is less than 5 ml, very low. And Hergy channel inhibition it is having plus indicating. Human hepatotoxicity it is having plus, Ames immunogenicity minus, and skin sensitization having negative. And LD50 it is having 2.291 at concentration 1767. So above 500. So it is a low toxic, acute toxicity. And drug induced liver injury. So having a liver injury, so this is omeprazole. And also we can find out this aggregated prediction also in the ADMET lab. So either it is aggregating or not. So this omeprazole, it is not aggregating. So it is categorized into 0. So it is probability of aggregation is very low. That is 0.06. So that is about the ADMET lab. So another tool in the ADMET. So pre-ADMET. So this link give that particular website. Pre-ADMET. So it is a web-based application. So to predict the ADMET, drug likeness prediction, so ADMET prediction and toxicity prediction, so as like in the previous databases. So these are the parameters we can estimate by using this pre-ADMET, all these values. And this tool that is pass online. So this need to log in. So it is going to giving the prediction of activity spectra for substances. So prediction of activity spectra. So various active probability to be active and probability to be inactive. So PA is indicating the probability to be active and probability to be inactive. So that data completely given to our search compound in that pass online. So here ibuprofen I given by taking this Smiles notation. So it given the activity spectra of the compound. So having the anti-inflammatory probability to be active anti-inflammatory 0.9 and inactive of anti-inflammatory that is 0.004. So not only giving the activity spectra, predicted activity spectra, and also it is giving the possible adverse and toxic effects of the compound. So for ibuprofen, so these are the possible adverse and toxic effects that is predicted, adverse and toxic effects. So this bleeding, occurrent bleeding is present for this ibuprofen. Then nearly it is having probability to be active 0.968 and inactive is 0.01. So likewise, the any molecule, if you are giving either in the form of Smiles notation or MOL form, mole form, so we can get the complete activity spectra and also adverse and toxic effects of the compound by using this PASS online software. So that is by the way to drug predictive services. And another tool that is ProTox, that is prediction of toxicity of chemicals. So this is the website link I represented here. So temporarily it is in not working, this website, it is under maintenance. So this is going to give this complete toxicity profile of the compound, like acute toxicity, organ toxicity, toxicological endpoints and toxicological pathways and toxicological targets it is giving. So it is representing the different classes in the toxicity prediction, class 1, class 2, class 3, class 4 and class 1, class 6. So class 1 is if the compound K means it is fatal if swallowed. The LD50 is less than or equal to 5. And class 2, it is fatal if swallowed, having a 5 to 50, LD50. And class 3, toxic if swallowed, and 50 to 300. And class 4, harmful if swallowed, 300 to 2000. And class 5 may be harmful if swallowed, that is LD50 is 2000 to 5000. And greater than 5000, it is non-toxic, class 6 category. And another software that is mole inspiration. So this website link, it is going to helpful. So by clicking this free online services in the mole inspiration, we can get the parameters like this. So I given the ibuprofen structure. I calculated, we can calculate this log p value, topological polar surface area, number of atoms present in the compound, and number of hydrogen bond acceptors, donors, and number of violations, that is according to the drug likeness properties, number of rotational bonds, what is the molar volume. So we can calculate by using this mole inspiration. And also we can find out this bioactivity scores we can find out. So either it is a G protein coupled receptor ligand or ion channel modulator or kinase inhibitor or nuclear receptor ligand or protease inhibitor and enzyme inhibitors. So these parameter bioactivity scores we can find out in this mole inspiration. So in this slide, I represented some of the In-Silico softwares or tools used in the different drug discovery process. So several tools are there. I represented some databases I represented in this slide. So these are the different databases and molecular modeling tools, drawing tools, homology modeling tools, docking tools, QSAR tools, screening tools available, and ADMT toxicity tools available. So some tools I represented, so several if you are seeing. So thank you.
Thank you, sir.
Hello, sir. Thank you, ma'am.
Yes, sir. It is valuable session from you, sir.
Ma'am, can I stop this sharing option? I need to
Yes,
keep.
sir, it is for session open for question and answer, sir.
Okay, okay, madam. Okay, madam.
Any queries from the audience? You may directly speak with the speaker.
So all our online tools, we can easily access all these tools except some tools like Schrodinger and Stardrop Direct Nexus. So remaining all online tools, so you can get the complete information of all these parameters in those websites.
Yes, sir. Sir, from audience, they request you to show previous slides, sir.
Okay, ma'am. Which one they ask, ma'am?
Last but one, sir. Before the
Last but
thank
one.
you slide.
Yes, ma'am. I'm going.
Yes, sir. Last one, sir. Before the slide of thank you, which cover all the freely available softwares.
Yes, ma'am. I'm moving. Yes, ma'am. Yes, ma'am. Moving. Yes, ma'am.
Yes, sir. Any questions from the audience? Raise your hand who have queries.
Ma'am, one Ashwini Misra.
Yes, sir. Ma'am, you may ask the question. Otherwise, you may text in the chat box, ma'am.
She's texting, ma'am, asking that